Okay, thank you everyone for coming. I think we'll start now. So I've taken over from Anna in organising the seminar series. So that kind of leaves me in the awkward position of having to introduce myself. I've met quite a lot of you now. I've been here for about a little over a year. So my name's John and I've joined the Plant Productivity Group as a lecturer. To give you a little background on myself, I'm from... Right, slide's not working. There we go. I'm from not far from here. I'm from a little village called Viewers, about 20 minutes away. That has the River Stour running through the middle of it. North of the river is Suffolk, south is Essex. I grew up on the north side, so I'm local, but I'm not a proper Essex boy. I went to university at UEA, where I got an undergraduate degree in ecology and a master's degree in crop improvement from the John Centre, both in Norwich. I then came here to do a PhD with Uli, where I studied water use genetics in Arabidopsis. It's a model plant species, and I'll be talking a little bit about that in my presentation. After that, I moved to the US. I did a postdoc at the University of Illinois on water use genetics and physiology in sorghum, important crop species. Came back to the UK to do another postdoc at Nottingham, looking at heat tolerance in rice. And then before coming here, I've been working at the University of Cambridge, where I've been studying the genetics and physiology of photosynthesis in important EU crops, mainly barley, tomato, and maize. But today I'm going to talk about largely unpublished work that I've done, looking at water use in natural populations and how they can teach us lessons on how to improve crop sustainability from a water use standpoint. So when we talk about plants and crops and productivity, we often talk about net primary productivity when we're looking on a large scale. So that's essentially the net gain in biomass per unit area and time. It essentially boils down to photosynthesis, so carbon gain, minus respiration, so carbon loss. And there's lots of classic ecological research studies that have shown that net primary productivity is largely a function of the availability of water. So this is a classic study where there's 11 or 12 different long-term ecological research stations across the Americas, where we can see that net primary productivity, let me put my laser on. Net primary productivity is largely a function of how much water is available. So that kind of highlights how important water availability is for growth and biomass accumulation in plants. And we can see that we can get further evidence from this from a kind of evolutionary perspective. So a lot of the features that characterize plants are a result of evolutionary adaptations to associate with water use and balancing water use with carbon gain. So you have things like cuticles on the leaves, which are lipid polymers that basically serve as a permeable barrier to prevent water loss. Tracy's work, which I'm sure you've heard a lot about, has talked about stomata. So these are pores on leaf surfaces and other tissue types that are flanked by two guard cells, and they allow plants to respond to environmental cues to balance water loss with carbon gain. Grass species have these things called bulliform cells on the leaves, which as they lose water, they cause leaves to roll up, which minimizes transpiration further. Again, it's a mechanism for preserving water. And then root system architecture has evolved largely to promote water uptake along with nutrient uptake as well. For example, root hair cells can increase their density to increase the surface area of the roots to the soil to promote water uptake. And even we can look at at the level of metabolism. So C4 photosynthesis has evolved independently in many different lineages of plants, and it's largely evolved in dry and wet areas, and it allows it affords plants a kind of an internal CO2 pump, so they can close stomata and not have any cost to carbon gain, but improve efficiency of use of water. So given the importance of water availability, then it's not exactly unsurprising that primary productivity is highest in and around the tropics. And that's reflective of the fact that that's where the rainforests are and that's where precipitation is the greatest. But those places, quite rightly, are not exactly where we grow most of our food. And you can see that from this figure here. So this is basically showing net primary productivity, but the human appropriation of that net primary productivity. And you can see we grow a hell of a lot of our food in places where it rains comparatively less than the tropics. It still rains enough to be able to grow food, but nowhere near as much. So this is a problem for a couple of reasons. Firstly, we're not producing enough food right now, and it looks likely that we're not going to be able to produce enough food in the future. So this is this figure here is showing the increase in kind of harvestable yield of 4 of our most important crop species since the Green Revolution in the 1960s. So you can see in all 4, we're basically on a linear trajectory in our ability to produce harvestable yield from these crops. And that's a result of breeding. So plant breeders are crossing different varieties together to get more productive lines. We're not growing the same variety of wheat, let's say, in the field today that we were 10 years ago. It's new and improved with better genetics. But it's also a function of improved agronomic practices. But if we extend those trajectories out into the future, into 2050, you could just through a linear regression, we're not going to be able to produce enough food for our growing population. So future food security is at risk. Whereas where we really need to do is start increasing this way. So have a more exponential increase in our ability to produce food. And secondly, that's a problem because the areas where we grow a lot of our food, you look at this human appropriation of natural primary productivity, you pay attention to the darker areas, and then you look at this figure here. This figure is showing us if we follow the kind of middle of the roads IPCC carbon emissions guidelines, we're still going to have a massive reduction in the amount of precipitation in key crop growing regions. So if you look at Europe, California, Mexico, these are all really important areas where we grow a lot of the food that we eat. And the color scale here is basically showing the predicted difference in precipitation in 2050. So in 2050, we're looking at 10 to 20% reduction in the UK, and even more in France and Spain, where we have important regions growing crops. And I think it's really important to see these kind of data and to try and understand the problem. But you can kind of conceptualize it even more when you see things. So last year I was in Italy on a project to characterize physiological variation in photosynthesis in tomato, in field trials in tomato. And we were doing that in a region around Bologna and Parma, which is kind of the breadbasket of Italy. It's where they grow a lot of wheat for pasta. It's where they grow a lot of rice for risotto. And the water that's there to promote that growth extends out from the Po River. And this is the Po River through the middle of Parma last year when we were doing that field trials. It often dries up, but this is an absolutely extreme example. And actually, when we were doing that last year in Italy, there was the state declared a state of emergency because there was so little water, and that was really impacting agriculture. So I think it's important to kind of really visualize that and understand how much of a massive problem that is. So in terms of what I'm going to talk about today, I'm going to talk about 2 case studies where I've looked at natural populations and how water use varies within those populations, and how the lessons we learn from those can go forward to an informed approach to try and improve the sustainability of water use in crops. So the first one, I'm going to talk about how water use is under selection in Arabidopsis thaliana. So Arabidopsis thaliana is kind of the lab rat of plant biology. And one of the other really nice things about Arabidopsis is there has in maybe 10, 15, 20 years ago, there's been a huge surge in collecting wild varieties of Arabidopsis from all over the northern hemisphere, and we have great genetic information. They've all been genotyped and sequenced, and we have excellent information on exactly where they come. And it's a really tractable species to work with. You can grow it quickly. It's got a small genome. It's a great species to work with. So this is all work that I did for my PhD that I haven't actually published yet because I got to reanalyze a lot of the data when I had downtime during the COVID pandemic, but I'm hoping to get it out soon. So one of the experiments I did with Arabidopsis is I took hundreds of different varieties of Arabidopsis from all over the northern hemisphere, from really different environments, dry environments, wet environments, hot environments. So they've evolved, their adaptive basis is very different because they've had different environmental challenges. And what we did is these experiments where you have them in pots that are really well soaked, so they've got plenty of access to water, and you let them dry down. So that's what you can see here. So we have two different varieties here of Col 0 and SEO. So the individual dots are different plants representative of this different ecotype. So we have Col 0, which is the reference genotype, and SEO, which is a genotype from Spain and an arid environment. And as you can see, as you progress forward in time and don't give these plants water, the relative soil water content, so how wet the soil is, dries down progressively. And this relationship is slightly curvilinear, but it is, you can fit a linear model to it. And that linear model is, we take that as a metric of water use, and we call it vegetated water use. And we also measure a bunch of other parameters to do with life history and phenology of these populations, and different water use parameters. And when we do a kind of clustering and correlative analysis between all of these parameters, you can see that all the traits that are associated with water use, they cluster really far away from the phenology and the life history traits. So suggesting that water use is independent of those key features of the plant life cycle, basically. So we can assess that independently and not be worried that the traits to do with water use are just are due to variation in other parameters. So what we really wanted to do, we're growing all of these Arabidopsis populations under controlled environments, but what we want to do is look for signals of climatic adaptation. So one of the first experiments we did to see if we could do that was try to grow Arabidopsis in controlled environments and environments that are more reflective of natural settings. So we grew them outside. That's something Arabidopsis researchers would never normally do. But what we wanted to do that to see if when you grow a plant in a really pampered environment, do you get similar results if you grow it in more natural settings? And the answer was you do. So we focused on 3 parameters. So we have these axes of labels need to be fixed. But basically, on the x-axis, you have 3 traits measured in the outdoor, and on the y-axis in the indoor. And the traits we looked at was basically total biomass, so how much biomass the plants put on during their life cycle, highly correlated between the 2 environments. The seed biomass, so that's a metric of reproductive fitness, right? It's an important component of ecology. So that was really correlated between the 2. And then this parameter here, which is delta carbon 13. So delta carbon 13 is the ratio of the 2 abundant carbon isotopes in the atmosphere within the tissue of the leaf, so carbon 12 and carbon 13. And that is a really good indicator of water use within plants for reasons I don't have time to go into now. But again, this showed us that water use in the indoor plants is similar to plants grown in more natural settings. So our experiments that are very controlled are reflective of how plants might grow in the environment. So we can use the subsequent data that we generate from those experiments to look for signals of climatic adaptation. And to do that first, what we wanted to do was we're collecting a lot of data on lots of different traits, so we want to reduce the dimensionality of that. So we did principal component analysis. So these are all our different populations. These are all the different traits we looked at. And we got three significant principal components from that analysis. And the one I want to focus on the most is trait principal component 2. So you can see these large circles here, they're essentially the correlation coefficient to the principal component for the different traits. And you can see trait principal component 2 is primarily defined by these 4 parameters here, which are all to do with water use. So the first 3, vegetative water use, slope 1 and slope 2, they're all to do with how much water they're using. And the break point is essentially due to drought sensitivity. So as I mentioned before, we have all these different ecotypes and we know where they're from. Because we know where they're from, we know the latitude and longitude where they were collected, we can use that data to query bioclimatic databases to get climate information. So all these numbers here, bio 6, bio 11, bio 9, bio 1, they all refer to different climatic parameters like temperature, precipitation, and throughout the year, and things like climate stability. So we did a principal component analysis on that data as well, and to generate principal components. The one we're interested in is principal component 1. And I know a lot of these numbers probably don't mean anything to you, but I can tell you that principal component 1 is mainly, climate principal component 1 is mainly defined by climatic parameters associated with precipitation. So what we were able to do then is compare the trait principal component scores from these populations with the climate principal component scores to look for signals of adaptation. And one of the associations, the one that popped out the first was a positive association between climate principal component population score 1 and trait principal component population score 2. So as climate principal component increases, this is reflective of increasing temperature and reduced climatic variability. For the trait principal component 2, as that increases, that's reflective of increasing water use, vegetative water use, and increasing drought sensitivity. And this correlation might not look particularly strong to you, but when you consider it's collecting data from plants from all over the northern hemisphere, where there's a lot of variation in climate, is actually a pretty strong signal in that for these kind of analyses. And we could provide further evidence here by taking a genetic clustering approach. So we have genetic information, so SNPs, single nucleotide polymorphisms, for all these populations, and we did a clustering analysis. So for all the populations that we measured, we did a clustering analysis, which resulted in all the populations being grouped into 3 clusters. And because of the relationship between genetic distance and geographic distance, they tended to be populations that were close to one another. So you have 4 that are reflective of the distribution of Arabidopsis across the northern hemisphere. But then if we compare mean temperature and water use across these 4 clusters, you can see that cluster 1, high temperature, high water use. Cluster 4, low temperature, lower water use. So this is again reinforcing the association between temperature, the temperature at the site where these populations have evolved, and how much water they use. So water use is exerting a selective, temperature, is exerting a selective pressure to promote water use, presumably to afford thermoregulation. So the more water the plants use, the better they are at being able to cool down. So we were able to take that vegetative water use data and perform, again, using that genetic data that I just described for the clustering, to perform association mapping. So this is genome-wide, the summary of a genome-wide association study. So Arabidopsis has 5 chromosomes, and each of these individual dots represents a different single nucleotide polymorphism, so a mutation or a polymorphism across the 5 chromosomes. And when the dots are higher, that means the association to that particular area of there's an association between that particular area of the genome and the trait that you're interested in. So when we do that, we find 2 single nucleotide polymorphisms that go above a significance threshold. So these 2 these two SNPs, which represent areas or very specific positions within the Arabidopsis genome, are associated with variation in vegetative water use. And these two SNPs are within right next to or within linkage disequilibrium of just one gene, and that gene is a MIB transcription factor, MIB59. And just when we were doing this, there was two papers came out at similar sort of time, which implicated MIB59 in being involved in stomatal conductance and root growth. So if you remember, we're talking about water use, and we've identified a gene that's clearly involved in stomata, which balances with stomatal activity, which balances water use with carbon gain, and root growth, which is important for promoting water uptake. So we've had pretty high confidence that this gene is probably involved in water use. The other thing we were able to do is take, so we just looked at a subset of all the populations that have been genetically characterized in Arabidopsis, but there are actually thousands. So what we can do is query this specific gene, MIB59, for functional polymorphisms within the gene that are predicted to have an effect on the protein that it codes for. And across all the thousands of Arabidopsis populations that genetic information is available, we find 5 polymorphisms that exist and that are predicted to have a detrimental effect on the protein structure. And one of the things we did was we compared the different alleles, so the different varieties of these different genetic polymorphisms. We compared those to temperature, climatic parameters. So one of them was mean annual temperature, and you can see that specific alleles are separable, can separate when you compare the temperature from where the population arises from. And this actually showed us the functional alleles of MIB59 are way more common in warm environments. So in warm environments, there's selective pressure to have a functional MIB59. And just to show you, we compared all the different climatic parameters. So the yellow ones are associated with temperature, the green ones are associated with seasonality, and the red ones are associated with precipitation. And these are the different polymorphisms here along the side. And overwhelmingly, there was a significant interaction between the alleles of those polymorphisms and temperature, but we didn't see that with precipitation. So again, this is reinforcing the idea that MIB59 is associated with water use for an adaptive purpose. And as I mentioned, previous studies have mutated MIB59, so they've knocked it out with CRISPR. We asked the authors if we could have those seeds. They gave them to us, and we did experiments where we grew wild type, so they have a functional MIB59 with induced non-functionality in MIB59. If you grow it under low temperature, you don't see much difference in water use. If you grow it under high temperature, a functional MIB59 is clearly important for promoting water use for thermoregulation. And we also looked at water potential, so that's kind of indicative of how water stressed plants are. Again, under low temperature, no real difference, but under high temperature, if you don't have a functional MIB59, your water potential decreases, the plant is stretched out. And these results have kind of shown us that anatomical features, namely stomata and root length, which we verified, are under direct selection in specific environments, and the associated functionality of this associated gene is key to maintain optimal plant function. So on to the second case study. So modulated gas exchange under selection in Hordeum spontaneum. So that is the Latin name for wild barley. So Hordeum bulgari is cultivated barley that used to make beer and for other purposes and animal feed. Spontaneum is wild barley. It exists or originated in the Fertile Crescent in the Middle East, and that was selected for to produce over millennia to produce the cultivated barley that we have now. So this, unfortunately, we're all familiar with this area of the world now. This is Israel, and we have 320 different varieties of Bordem spontaneum that were collected from this area. And we've grown them out under several field trials now in a common garden experiment in Cambridge. And we did this to assess variation in photophysiological traits, so traits to do with leaf level gas exchange and photosynthesis. And this has been gives me a nice opportunity to share this cool video we did over the summer. And eagle-eyed among you will see this isn't barley, this is maize, but we have our maize field trials and barley field trials running side by side. And we, I need to get rid of the points. I can't do it. Anyway, we have these plants growing in the field. We collect leaves from them. We bring them back to the lab. We run them through this phenotyping pipeline where it assess loads of different traits related to photosynthesis, carbon metabolism, and leaf structure. And we use that to generate data that we use for essentially genetic mapping to try and identify traits that are associated with photosynthesis. And you'll see that we're measuring leaves that have been cut. And we've recently published a paper that shows that if you measure leaves that are cut, the data that you generate is exactly the same as if you measure leaves that are attached. So there's no issues with that. But back to the population that we're working with. So we're working with this wild barley variety. Again, like with the Arabidopsis, we have genetic information, although not as much, but still enough to do some work with. And what we can do kind of a structure analysis where we take all that genetic information and we cluster the different varieties, and that allows us to cluster them into distinct groups. And these groups, again, like with Arabidopsis, they cluster into areas of adaptation. So you have coastal varieties from the southern desert, the eastern desert. And then we can take some of the parameters and the traits that we've measured to compare how they are different between the different evolutionary groups of this wild barley. And the genetic groups show us what was really nice. The first thing we saw was really expected trends in phenology. So these two graphs here are showing differences in days to heading. So that's flowering time essentially. So how quickly the plants flower, and also specific leaf area. So specific leaf area is part of the leaf economic spectrum. So it's basically the ratio of dry mass of the leaf to the area. And that's a really easy trait to measure, but it's really indicative of a lot of interesting things like growth rate. It's very highly correlated to the rate at which plants grow. So one thing you can see here is that plants from the eastern desert, where we presume it's probably more dry, or we know it's drier, more arid, these plants they flower much earlier, and SLA is negatively correlated to growth rate, and their rate of growth is much quicker. And that's because they're in a dry area. They need to put reproductive product out really, really quickly to promote their fitness. So they need to basically get from seedling to seed as absolutely quickly as they possibly can. Otherwise, they're likely to succumb to some kind of stress. And that's not the case in varieties that are from areas that are less arid where there's more water available. So this is commonly known as a drought escape strategy, but that we saw a really obvious trend of that in our common garden experiment. And the other thing we were really interested to see was very obvious patterns of stomatal constraints to photosynthesis. So in this graph here, you'll see stomatal conductance to water. So that's essentially a measure of the rate at which water is leaving stomata, and that's proportional to how open they are. So in a dry environment, you would expect stomatal conductance to be lower because there's less water available, so the plant wants to conserve it. But in a wet environment, there's plenty of water available. They want to keep their stomata really open so they can maximize how much carbon goes into those pores for photosynthesis so they can grow. And we saw, again, a nice trend here. So in drier environments, stomatal conductance is much, much lower than in other environments. This is an adaptive signal. And another trait, so SL, this is stomatal limitation. So this is basically modeling the extent to which the behavior of the stomata are impacting photosynthesis. So we can see, again, an obvious trend. Here we're in desert varieties where stomatal conductance is reduced, you have more, that they're limiting photosynthesis more. So what we wanted to do was kind of a deep phenotyping experiment where we selected particular varieties at random. Well, we selected varieties at random that come from two adaptive groups to try and verify those results and look for other signals. So we picked varieties from the Eastern Desert and coastal varieties. And what you can see if we zoom in on exactly where those varieties are coming from, you can see these varieties. So we had varieties from the coastal areas are clearly water availability is higher compared to the desert varieties, which look exactly like a desert, but somehow plants are still able to grow there well. So what we did was we did these experiments where we had these different varieties, 4 from the coast and 4 from the east, and we grew them under different watering regimes. So some of them we allowed them to constantly have 40% of the soil soaked, and some of them we allowed to have 80% of the soil soaked. And we assessed photosynthetic efficiency and life history traits throughout their life cycle. And we saw, again, similar results, but allowing us to tease the differences out a little bit better. So in these plots here, you can see the individual varieties, but we've grouped the data into desert and coastal here. So this is stomatal conductance. So again, the rate at which water is leaving the stomata. And you can see if we compare 40% water availability to 80%, in the coast, we see a much stronger reduction in this trait in coastal varieties than we do in desert varieties. And that kind of translates into total biomass as well. So you can see if you grow a coastal variety in a dry environment, which it's not used to, it has a much greater reduction in total biomass that it puts on. So measured as total leaf area here, compared to the desert variety, which is adapted to a drier environment. So this is actually really, we've still got to dig into this data a little bit more and do a few more statistical analyses, but this is really exciting. This is one of the first kind of accumulation of evidence that shows empirically that modulated gas exchange is contributing to plant adaptation. So it's quite exciting, but we've got a little bit more work to do to kind of polish this up. So I kind of hope with those 2 case studies, I've convinced you that stomatal morphology is really important for balancing water use and carbon gain in plants. And so now I want to go into more of like previous experiments were more kind of not fishing expeditions, but trying to see what was there and trying to accumulate evidence. Now we're in what I'll talk about now is more a targeted approach to try and identify genes of interest that could associated with stomata and transgenic approaches to try and improve water use efficiency in sorghum. So sorghum is a really important crop species, not so much in Europe, but in the US and Australia, it's closely related to maize. So one of the things, so this is all work I did on my postdoc in the University of Illinois. So I helped to kind of develop this microscopy method using optical topometry to characterize 3D leaf surfaces. So looking at stomata is notoriously difficult. The gold standard approaches for doing this are quite complex. So we developed this method where we could use these microscopes and just put a leaf directly underneath them, no sample preparation, nothing, and generate this 3D reconstruction of the leaf surface, basically. And you can see the stomata here. So this is the pore here, and these are the individual guard cells here that increase and reduce their turgidity to open and close the stomatal pore to let water out and carbon in. So this really broke ground in terms of measuring a trait that was really slow to measure to make it into a high throughput protocol that could be measured quickly and easily. And we collaborated with computer scientists to develop machine learning approaches to automatically count how many stomata were in these fields of view. And that's what you can see here. So we get a really nice correlation between the number of stomata that are there if we count them manually compared to the algorithm that was developed. This is actually quite poor now. So there are new approaches that have come out that are way, way better and get much higher levels of agreement. But the main thing for us was we wanted to get a relationship that was positive, so we could use that data to do genetic mapping to try and identify new genes associated with stomata, this important crop species. And this is my colleague who I was working on this project with before, Caitlin Moore. She is now at the University of Queensland. No, Western Australia, sorry. And she's standing in front of the sorghum that we worked with. So it's actually bioenergy sorghum. It's absolutely massive. You get 15 to 20 feet tall. They're all varieties from Africa and they're photoperiod sensitive. So they need a particular day length to flower. And if you grow them in the Midwest in the US, they don't get that day length because the days are much longer to what they're used to. So they just keep growing and growing and growing until winter falls, and then they will maybe eventually flower. But the other thing, I'm actually going to skip this in the interest of time. So we generated a lot of data on stomatal density and stomatal patterning across these varieties. And we used that to do, again, genome-wide association mapping to look for signals across the genome that are associated with stomatal patterning. And we also, with our collaborators at Cornell University, pioneered this new approach called transcriptome-wide association mapping. So before we're looking at genetic signals, now we're looking at expression signals. So we had RNA-seq data, so transcriptome data from all of these different varieties. And here we're looking at genetic variants and how they're associated to the phenotype. But here we're looking at how expression variation. So each dot in this figure is associated with a genetic polymorphism, but here each dot is associated with a particular gene and its expression. So it's correlating expression variation with the trait variation. And then we can combine these results with a Fisher t-test to generate evidence from two sources for genes that might be associated with stomatal density. And we identified many. And what was most pleasing was the one that we identified as second most significance was mute. Mute is one of the most well-characterized stomatal development genes. So we knew that our approach was working. One of the other genes we identified was EPF2. EPF2 is also really well associated with stomatal development. At the same time, our collaborators at the University of Nebraska were making transgenic plants which had mute knocked out. And that allowed us to develop lines which had reduced stomatal density. EPF2, sorry, not mute. And then when we grew them under a water withdrawal period, we can see that lines that have reduced stomatal density are able to maintain photosynthesis under this water withdrawal period for longer compared to lines that haven't got that mutation and that have higher stomatal density. So we had developmental issues. So this gene is clearly involved in other processes. So that's why the plants look a lot more bushy, but still it was preliminary evidence that showed us that manipulating stomatal density genes that we identified for this cool approach that was afforded by the new phenotyping method we developed allowed us to identify genes that can be targeted to improve water use efficiency of crops. So not quite done, but just to acknowledge the people involved in the work that I did. So I did the Arabidopsis work under the supervision of Uli when she was here as my PhD supervisor, and Tracy and Phil were involved in that work as well, helping along. The barley work I did at Cambridge under the supervision of Anna Cromdike on a project that's a collaboration with Tracy and Christine. Aniel Friedman at the Volcani Institute in Israel provided all the germplasm for that. And the sorghum work I did with my postdoc supervisor in Illinois, Andrew Leakey, and also colleagues there, Pat Brown and Sam Fernandez. Tom Clemente at Nebraska was responsible for generating the transgenic lines that we had. And computer scientists at Wisconsin helped with the stomatal counting, and colleagues at Cornell were involved in the quantitative genetics. So then just finally, this is all research that I've done in my previous life before joining Essex or rejoining Essex. But I'm now really fortunate to have acquired a little bit of funding to start doing some research here now. And although we're really only just kind of getting going, I thought I would share with you some, like, what we're planning to do and how we're looking to move forward. So I've been working with new collaborators and friends at the Beth Chatto Garden. Those guys have come today. It's nice to see you. Thanks for coming. So we're hoping to develop a smart irrigation system to minimize water use in their activities. So this is showing how, so Beth Chatto Gardens is just up in Elmstead. It's a beautiful garden, and they also a nursery where you can buy plants. And they, their nursery is stocked with plants that they propagate from their garden. So they take cuttings from other plants and they put them into these pots, into these trays to grow them up. And their irrigation system at the moment is through misting from these units here. And it's triggered through this kind of artificial leaf that when it dries out, electrical signal is cut off, and that stimulates the irrigation. But what happens is the plants end up being watered way more than they probably need to. So they're always really soaked, and this actually might be detrimental to them. So what we want to do is develop a system that use these relatively cheap soil moisture sensors that measure conductivity between these two prongs that fall into the soil or that go into the soil, which is proportional to how much water is in the soil. We want to use those to stimulate the irrigation. So when that conductivity falls below a threshold, a signal is relayed which stimulates the irrigation. But what is optimal soil moisture content? To do that, we've got 3 of their most popular species that are also popular plant varieties that they sell that are also quite drought tolerant. And we're doing an experiment here at Essex where we will be growing them at 4 different relative soil water contents throughout. And we will be assessing plant health by different technologies that we're really fortunate to have here at Essex as part of the Essex Plant Innovation Centre that Tracy has built up. So we'll be looking at photosynthetic efficiency, chlorophyll content, and water use, and also final biomass production and total water use outlay. And we'll use that information to basically identify what is the optimal soil water content to grow these cuttings at. And then this will kind of feed into a system where we have these moisture probes within pots that are growing the cuttings, and that will feed into a controller which is connected to an irrigation system. And when watering falls below a soil water content or when the relative soil water content threshold is reached, it will stimulate the irrigation. And I'll skip that slide. So the longer term outlook for that is we want to test the irrigation system at the optimal soil water content that we define based on our experiments that we're doing here at Essex at the moment, or we'll start very soon. And then we're going to use flow meters to test how much water is saved by these 2 different methods, so the old method and the one that we developed. And this is a project I have in collaboration with Hussein and Fahid in the School of Computer Science and Electronic Engineering. And they're going to help to kind of streamline the system and maybe develop an app where things can be monitored remotely, and you can select a specific target relative soil water content or calibrate it to different soil types. But this is like a cool thing to be involved in, and maybe it's kind of got impact case associations maybe later on down the line. So thanks to Ben, Megan, Joe, and Julia at Beth Chatto for being involved in that, and also to the acknowledging the Impact Accelerator Fund that Tracy has that helped to fund this project. And also working with these guys at Beth Chatto has also resulted in me having a fully funded studentship that's for Advert now to look to go and work in their gardens and to do comparative phylogenetics of the leaf economic spectrum in the amazing diversity that they have up there at their gardens. So thanks for supporting me for that, guys. And then that's all I really had to talk about today. So finally, thank you for your attention, and I'm happy to answer any questions that you might have.